He's the only one who is still walking feet, right? Walking feet and alive and kicking here. And before I bring in, I need some of your collaborations with that, can I? Oh, I like that. That is the culture, I like that. Oh, okay, first and foremost, I like to trade in applause, can I? Come on, 1, 2, 3. Macam not enough lah, cukup deh. Oh, okay. Without further ado, let us put a big welcome.
Go block the way, please. Thank you.
And welcome the speaker for the day, Nobel Prize winner, Dr. Ferid Murad. And our father of BE, Mr. I.R. Lee Xuesen. And also the founder and executive director of International, Miss Ho Wei Ji. Okay, once again, we welcome Dr. Ferid Murad in the house so they're at Suntec City Convention Center, the Nobel Prize laureates of this pharmacology. All right, could everyone take a seat? The event is going to get started. Yep. Yeah, before I proceed, I hope you appreciate that today is the day that everyone, you come in here and you paid for the attendance here. So we appreciate no live recording, no FB live, as it is going to be live by our own company, BE International, and is going to post at the FB. All right. So for those who have that live when the event started, I'm going to have to get your handset to be as a lucky draw. Can I? Can I? All right. Very good. Okay, as for today, as for the opening, we are going to show you exactly what is it all about for today, which is the studies and all the studies and surveys by Dr. Ferid Murad. Let us present to you the video presentation as the opening.
And the winner is...
And the winner is... Gosh, what's with all the award shows?
The winner is... Congratulations. And I know to see the Nobel Prize.
Nobel Prize? What's that? Beats me.
What? You don't know what the Nobel Prize is? We're going to have to do something about that.
Whoa! What's up with this? Yeah, sure. Why not? So, what's your story?
Well, my name is Ferid Murad. You call me Fred. I'm a doctor and a researcher and scientist at the University of Texas Houston Medical School. And oh, by the way, I got the Nobel Prize in Medicine in 1998.
So, what is the Nobel Prize, anyway?
Well, the Nobel Prize is one of the greatest awards you can get in the world. It's recognition from other scientists.
So?
Well, you get to be on TV all over the world. There's a big party in Sweden. You even get to meet the king and queen of Sweden. You get a gold medal, and then of course there's the money.
Money? Royalty?
Yeah. Maybe it'd be better if I show you. May I?
Sure.
Thanks.
Oh, thanks, Fred. Welcome to my world of science and my laboratory. You know, the Nobel Prize wouldn't even be around if it weren't for...
Alfred Nobel, the sweet businessman who created the Nobel Prizes, was the guy that created dynamite. Nitroglycerin is the explosive chemical in dynamite. And even though it was very dangerous, Mr. Nobel figured out a way to contain nitroglycerin so that it could be put to good use, like to build stuff.
You could say his discovery rocked the world. But nitroglycerin has other uses. When Nobel started having heart problems, his doctor actually prescribed nitroglycerin for his heart. But Nobel said,
No way!
so he blew it. He figured anything that could blow stuff up can't be good for your heart. Nobody knew why it worked, but it did.
100 years later, Dr. Murad stopped to ask why nitroglycerin helps chest pain. And he shared the 1998 Nobel Prize in Medicine with Dr. Robert Furchgott and Dr. Louis Ignaro for figuring it out.
So you're the dude that figured out why nitroglycerin helped people's hearts?
Well, yeah.
So?
So what?
So why does it work?
We were trying to answer the question as to how nitroglycerin works to help with chest pain. I did experiments, observed the results, and collected data. Then I found out if what I thought was right or wrong. Anyway, what I did find was that nitroglycerin releases nitric oxide, and that nitric oxide does a lot of important stuff in the body.
So what is nitric oxide, and what exactly did you figure out about it that got you this Nobel Prize?
Let me show you.
It protects the heart, it stimulates the brain, it kills bacteria, and it's a real gas. It's nitric oxide. No one can say no to NO.
Nitric oxide, or NO, is a simple molecule with two atoms, nitrogen and oxygen. And yes, it's a colorless, odorless gas. A scientific sensation sweeps the globe. Nitric oxide is everywhere. This common and toxic pollutant depletes the ozone layer. It's even found in car exhaust and cigarette smoke. But from supermenace to superhero, nitric oxide is also found inside the human body and can help send very important messages, which are not from our sponsors. When blood flows through your blood vessels, the inner lining, or endothelium, releases nitric oxide. The nitric oxide signals your blood vessel to relax and widen.
So what?
This in turn lowers blood pressure, the force which the blood exerts on the vessel walls. If your blood vessels make enough nitric oxide to signal your blood vessels to relax, then your blood flows on through no problem. But if blood doesn't flow through, blood clots form. Then...
And that's not all.
So, relaxed blood vessels allow more blood to flow, and nitric oxide can have an impact on all different parts of the body.
For example, nitric oxide is already saving the lives of babies who were born too early. By breathing in very small doses of this gas, it helps their lungs and improves their breathing.
And that's good.
In nerve cells, nitric oxide can stimulate the brain, affecting things like behavior.
Oh, behave, baby. As part of the body's self-defense mechanism, nitric oxide defends against tumor cells and bacteria too.
It's amazing stuff.
But nitric oxide is no laughing matter, and not to be confused with nitrous oxide, better known as laughing gas. Somebody turn off that gas.
So how do you think of all that anyway?
Well, over time, I became very interested in how cells talk to each other. But most other scientists didn't think that was very important.
Dr. Murad figured out that when cells talk to each other, it's like one cell sends an email to another cell somewhere in the body. And the email is the gas nitric oxide. The email can break into another cell and take over how the other cell works.
It may contain a message, like instructions for a blood vessel to relax.
Or it may contain some other type of instructions. For example, if the message is being sent to a cancer cell, the nitric oxide may kill the cancer and then self-destruct. Hasta la vista, baby. Nitric oxide in your body affects so many things. It's like having a worldwide internet system inside your own body.
So why did you go into science in the first place?
Well, it's really a lot of fun to figure out how stuff like this works. And you don't have to be brilliant to get ahead. You just have to have some goals and be prepared to work very hard.
As a scientist, you get to do something for the first time that nobody else has ever done, and that's exciting. Sometimes your discovery opens the door to a whole new way of thinking, and even more new discoveries. It's really cool. It's kind of like the Science Olympics, and the gold is the Nobel Prize.
Teams of scientists around the world compete with each other. It's fun. Who's going to finish first? Who's going to win the prize?
One of you could be a Nobel Prize winner someday. Who knows?
Hey, I had some more questions.
Yeah, me too.
Well, I guess we'll have to find out more on our own.
Maybe we could check the internet.
Yeah, and look up science and the Nobel Prize.
They're gone. They left some popcorn, didn't they? Yum.
Sorry. Okay, that is the video presentation specially arranged for everyone's view for today. Okay, as you can see on the video we have shown just now, there is the honorable gentleman who is there being involved in that interview. He is the man we have been waiting for. But before I bring him on the stage, I would like to tell you a little Department of Medicine, Stanford University, and also the Senior Advisor at Stanford Veteran Administration Hospital. In addition to being the Nobel Prize recipient, Dr. Ferid Murad has received numerous awards and honors, including the Albert Mary Lasker Basic Medical Research Award, American Association CBAR Award, Baxter Award for Distinguished Research of the Biomedical Science and Associations of American Colleges, and received more than 20 honorary degrees and numerous honorary professorships, and a member of many universities and society. Without further ado, let's put up a big warm welcome our Nobel Prize laureate, Dr. Ferid Murad, accompanied by our founder of BEFIL, Li Xuesen, on the stage for Dr. Ferid Murad's talk.
Well, thank you very much. When I learned that I was getting the Nobel Prize in 1998, I was chairman and professor of a department at the University of Texas. And many of the local schools, high schools, the middle schools, colleges, invited me to come and talk to the students. And I did quite a bit of that. It was a lot of fun, I enjoyed it. But I was very, very busy with a large department, a lot of invitations to travel and give lectures, and a big laboratory doing research. So I was pretty busy. And I decided I was going to make a video to reach out and influence young people to get them interested in science and tell them about the Nobel Prize. That was the purpose of this video. I've passed out CDs all over the world. I travel everywhere around the world. And I pass out these videos to influence young people and students. And it's been very, very important and exciting. The field of nitric oxide research has absolutely gone bananas. We discovered nitric oxide, and I'll tell you a little bit about how that happened, in 1976. We published our first publications in 1977. Today, 40 years later, there are 160,000 research papers on nitric oxide. Pretty remarkable. And it influences almost every area of biology. And when I travel, people say, I work with such and such an enzyme or pathway in biochemistry. Do you think nitric oxide will influence my research in this pathway that I'm working on? And I always say, certainly. Now, I don't know if it will or not, but I can be right 90% of the time, because it does everything. It's incredible. But after we discovered it, for 10 or 12 years, nobody wanted to believe it. Nobody would believe that a gas and a free radical could regulate the activity of an enzyme, and that's what we had discovered. And I'll tell you just a little bit about that, and it's gone in a variety of directions since then. And it's resulted in the development of many, many nutritional products, cosmetics, and drugs. And some very important drugs. Let me have the first slide. I can't see if it's shown up there. Okay. As we evolved and developed, we started probably in the deep crypts of the ocean, single cells. And we wanted to become multicellular. And we sort of got started near the crypts of all the boiling magma coming out of the earth with all sorts of toxic chemicals. And to survive, we had to adapt our genes in biology and biochemistry to survive in this horrible environment. And with evolution, that happened. And then we took these horrible chemicals and started using them as messengers to talk to other cells. So we can feed upon each other, colonize, and become multicellular organisms. I think nitric oxide, along with some other simple molecules, participate in evolution. So it goes back 4 billion years. Pretty remarkable. We know today that there are lots of messenger molecules in the body. And this slide summarizes that for you, the concept. There are 3 different cell types here on this slide, and they're all going to talk to each other. And I'm going to call cell type 1 a neuronal cell. It could be in your brain or a peripheral neuron in your fingertip or toe. It's going to produce a substance to talk to cell type 2. Cell type 2 I will call an endothelial cell, which lines all of your blood vessels. Every blood vessel and vein in your body has a thin layer inside called the endothelium. It's like an inner tube in your tire. And they're going to communicate with each other, and these neurotransmitters will regulate the endothelial cells, which in turn will produce substances to regulate the smooth muscle cells and other tissues nearby. And they do this by creating more molecules and messengers. So the first cell, if it's a neuronal cell, we call that messenger a neurotransmitter. If it's an endocrine cell, we call that messenger a hormone. If it's a macrophage or some other cells, lymphocytes, we call those messengers growth factors, cytokines, chemokines. So they have a variety of structures and a variety of names, but the concept is the same. It's a messenger to go talk to another cell. And when it's released into the bloodstream or the synaptic space, it identifies its target cell by the presence of a protein in the membrane of that cell. We call that protein in the membrane a receptor. And only the right messenger recognizes the right receptor. It's like the email in the video that goes only to the right address, right? So only the right messenger interacts with the right receptor only interacts with the right messenger. That's how the specificity of communication takes place. And while you have lots and lots of molecules and messengers, they only talk to the right cells that have these specific receptors. And when that interaction occurs in the cell membrane, it turns on a biochemical cascade inside of that cell to produce an intracellular messenger. We call them second messengers. The first intracellular second messenger, cyclic AMP, was discovered by my mentor and boss when I was a medical student and graduate student, Earl Sutherland. That was discovered in 1957. I joined the program in Cleveland as an MD PhD student in 1958, and I spent 7 years studying all kinds of hormones to regulate cyclic AMP production in a variety of tissues. It was really an exciting time to be involved in this fundamental research. My boss got the Nobel Prize in 1971 for discovering cyclic AMP. When I finished my training, I then took a residency in internal medicine at Massachusetts General Hospital in Boston. Learned a lot of clinical medicine. And then I went to NIH to work in the Heart Institute. And there I did more clinical medicine in cardiology and endocrinology. But I also worked in the laboratory studying cell communication and hormonal regulation of fat metabolism in fat cells. And about that time, another messenger was discovered in some tissues, in urine, and it was cyclic GMP, guanosine monophosphate. So while there were hundreds of these first messengers, there were only a handful of the intracellular second messengers. Cyclic AMP, cyclic GMP, calcium, diacylglycerol, the prostaglandins, so just a few. And they regulate all the biology and biochemistry of all these processes. So it's remarkable that only a small number of molecules regulate a lot of biology. And I worked with all of them, and that led me to figure out what cyclic GMP was doing. I knew hormones regulated cyclic AMP, and I thought perhaps hormones would regulate cyclic GMP, and I thought that maybe cyclic GMP would antagonize the effects of cyclic AMP. And I wanted to work on the biochemistry to figure that all out. And in the process, as we looked at the formation of cyclic GMP, we found some small molecules, azide, hydroxylamine, and sodium nitrite, would activate the enzyme guanylate cyclase to make cyclic GMP from GGP. I put these molecules on cell cultures, tissue slices from brain and liver and other organs. And I also developed a sample of tracheal smooth muscle from beef lung. And I thought that cyclic GMP would cause contraction of muscle because I knew as a student cyclic AMP caused relaxation, and I thought they would antagonize each other. So we put these molecules, azide, hydroxylamine, and sodium nitrite, on the tracheal smooth muscle in an organ bath. It elevated cyclic GMP as it did in other tissues, but the tissue didn't contract, it relaxed. Big surprise. And then I remembered as a resident When patients with heart attacks would come to the intensive care unit, we would give them nitroglycerin, nitroprusside, as vasodilators to lower their blood pressure so the heart didn't have to work so hard and would heal faster. So I told my trainees, let's put nitroglycerin and nitroprusside on the tracheal smooth muscle and see what it does. Does it cause relaxation as it does with blood vessels? And sure enough, it did. But it also elevated cyclic GMP. And in the course of early experiments in December of 1976, we realized that we now had some important drugs and molecules that regulated cyclic GMP formation. And I'll skip some slides here. And these molecules are summarized here to some extent. On the left-hand part of the slide are azide, sodium nitrite, hydroxylamine, and a bunch of organic and inorganic nitro and nitroso compounds, including nitroglycerin and nitroprusside. And they all activated guanylate cyclase, and they all caused relaxation. And we coined the term nitro-vasodilators. So that term has become popular. And the question was, how did they do it? How did they activate guanylate cyclase to make cyclic GMP from GTP, the substrate? Well, in the process of purifying these samples from various tissues, we figured out they were probably being converted to nitric oxide. And sure enough, when we generated nitric oxide by a chemical reaction, which is a pretty old reaction in your chemistry textbooks, we activated the enzyme. And that was the first time that a gas and a reactive free radical could activate an enzyme. It had never been done before. And I was very, very excited to figure out all the details of how this worked. So let me summarize for you as to its role in cardiovascular biology. This is a blood vessel with the endothelium on the left, the smooth muscle in the wall of that blood vessel on the right. And in red are 3 categories of vasodilators that will cause relaxation of that blood vessel to lower blood pressure. On the top are the nitrovasodilators that are precursors or prodrugs converted to nitric oxide. The nitric oxide activates the soluble isoform of guanylate cyclase. There are 7 different gene products that make guanylate cyclase, soluble in particulate isoforms. Pretty remarkable enzyme. Why so many genes? It's because each of those enzymes are regulated by a different messenger molecule. Either nitric oxide, atriopeptins, E. coli enterotoxin in the intestine to cause diarrhea. There are enzymes, there are hormones we've discovered in the intestine that regulate that enzyme to cause diarrhea and fluid transport in the intestine. We've found drugs that block that process that treat cholera and E. coli diarrhea. I've always wanted to do clinical studies. Whenever you have a natural disaster, whether it's an earthquake, a volcano, a tsunami, whatever is caused, the sewage system gets contaminated in the water system, everybody gets diarrhea. When Haiti had a hurricane, everybody was getting cholera. We have a drug that prevents that. From the biochemistry that we were doing, we discovered the drug. And I'd love to do some clinical studies to do that. I can't get any granting agency to give me the money to go off and do it. So I don't know, someday. But anyway, if you look at this pathway, the cyclic GMP made by nitric oxide from the nitrovasodilators, nitroglycerin, nitroprusside, regulates guanylate cyclase. That regulates an enzyme called cyclic GMP-dependent protein kinase that transfers the terminal phosphate from ATP to a protein substrate. The protein gets phosphorylated. And when that protein's phosphorylated, its activity, if it's an enzyme, goes up or goes down. And that in turn regulates calcium in the cell, and calcium is required for muscle contraction. And by lowering calcium, the myosin light chain and the myosin actin filaments that are sliding together apart to cause relaxation and contraction become dephosphorylated, you get relaxation. So over the course of several years, we figured out all the detailed biochemistry. And by doing that, you discover a lot of chemicals and drugs that can be applied now as new drugs for hypertension. Okay? We also learned, and this work came from another laboratory, it came from Robert Furchgott's laboratory, who worked at SUNY Downstate in Brooklyn. He was a vascular biologist, and we knew that drugs like acetylcholine and histamine in animals or patients would cause vasodilation, lower blood pressure. But in the laboratory, they never would cause relaxation of blood vessel segments of the organ mass. Something was wrong. What was wrong was as you made blood vascular segments, you destroyed the integrity of the endothelium because it's a very fragile cell lining. And if you're careful to preserve the integrity of the endothelium, these drugs will cause relaxation. And they do it by interacting with the endothelial cell on the left-hand side of the slide to permit calcium to enter the cell. Calcium regulates an enzyme that converts arginine to endothelial-derived relaxin factor, which is nitric oxide. So that's how arginine works, guys. Okay? And when he found that, he realized that EDRF, endothelial-derived relaxin factor, was very labile. I heard his work. I told him it might be nitric oxide. It might be a free radical and reactive species that was labile. And let's figure out how it's working. And we were going to collaborate, but we didn't. We moved to Stanford. He went back to New York. And later we discovered that EDRF is nitric oxide. Okay? And when you make EDRF or nitric oxide, it goes over to the smooth muscle compartment to cause relaxation, just like nitroglycerin. So it's an endogenous nitroglycerin. It's already there. Okay? Another category of drugs that work through this pathway are the atriopeptins. Atriopeptins are hormones first discovered in the heart, in the atrium. But now we know they're in other tissues. And they cause relaxation of blood vessels. And they also cause fluid excretion, sodium chloride and water, in the kidney. So they were natriuretic as well as vasodilators. So they shared the properties of nitroglycerin, but they also shared some of the properties of E. coli enterotoxin, which we found to activate one of the particulate isoforms of guanylate cyclase. We tested the atriopeptins, and sure enough, they activate guanylate cyclase, but another isoform. So this is basically how you regulate blood flow, blood pressure in all the animals in us. And it gives us a lot of clues now to how to treat patients who have heart attacks or strokes or whatever. And it turns out that nitric oxide not only dilates blood vessels, but it inhibits blood clotting, it inhibits aggregation of platelets, it inhibits atherosclerosis. It's pretty remarkable stuff. And I'm going to show you the list at the end where it does other things as well. Now, let me back up here. I think it's important that you see this slide. There are 3 different isoforms of nitric oxide synthase that make nitric oxide from arginine. L-arginine is the substrate for the enzyme. L-arginine is made in the body, but we don't make enough of it. So it's a semi-essential amino acid that we have to get in our diet or with supplements. We don't make enough. And young people who eat a lot of fast foods, and elderly people who don't eat the right vegetables and all, have deficiencies of arginine, and that leads to dietary supplementation with arginine compounds. That's why you're selling BEFIL. Okay? So we purified these isoforms of nitric oxide synthase. All tissues have them. Some tissues have more than others. They're very interesting enzymes. And they're very complicated with a variety of cofactors And cofactors are helpers. They're chemicals that are helpers to make the enzyme work more efficiently. Okay? So nitric oxide synthases have calcium, calmodulin. They have a heme prosthetic group. They use NADPH and oxygen and along with the arginine. And they use tetrahydrobiopterin as a substrate, as a cofactor to make this electron transfer to convert arginine to nitric oxide and citrulline. So, if you're going to regulate the production of nitric oxide, you want to supplement with arginine, and you want to reduce all of the reactive oxygen species that are getting oxidized with inflammation and atherosclerosis, so that cofactors will be active rather than becoming active. Instead of becoming oxidized, you want them reduced. That gives me clues as to how to create the formulas. And I've developed the formulas. You do L-arginine, various cofactors that are antioxidants. And today, the antioxidants are much more effective than they were 10 or 20 years ago. And a lot of the current arginine supplements out on the market don't have the right antioxidants. So the product we put in here in BEFIL is going to be the best product in the market, I think. And that all came from an awful lot of biochemistry over the years. So we did it. All right, so let me put the story together for you again. So various messengers, hormones, cytokines, whatever you want to call them, neurotransmitters, interact with their receptor and turn on this pathway of production of intracellular messengers. And these messengers regulate the activity of nitric oxide synthase, which makes NO, which activates guanylate cyclase, makes cyclo-GMP, which activates a protein kinase to phosphorylate proteins. That's the sequence of the pathway. And every biochemical step along the way becomes a molecular target for me to go screen chemical libraries to find new drugs. And that's what I do. So I take biochemistry to discover new pharmaceutical agents. There's a disorder called endothelial dysfunction. Patients with hypertension, diabetes, atherosclerosis, tobacco use, and perhaps obesity have blood vessels that don't make enough nitric oxide. And we figured out the problems with those blood vessels as to why they don't make enough nitric oxide. They make another arginine analog that becomes an inhibitor of the nitric oxide synthase. They also have reactive oxygen species. They make free radicals that oxidize the cofactors that are required for the activity of nitric oxide synthase. So that's endothelial dysfunction. So we now know how to treat that problem. How can we make diabetes better with insulin therapy or other agents? How can we improve the therapy of hypertension, atherosclerosis? So all of those new avenues for nitric oxide research have been very exciting to see that happen. And I'll summarize a little bit of that towards the end of the talk. Okay, here are 2 slides now, giving you sort of a quick overview of what cyclo-GMP and nitric oxide can do. It's a neurotransmitter. There are nerves in your brain, your GI tract, and your penis that release nitric oxide to cause vasodilation and have other effects. Okay? It regulates the turnover of fluid in your eye. It participates in glaucoma. Too much nitric oxide can be cytotoxic and destroy the retinal cells in your retina. Babies born prematurely have lungs that don't dilate their blood vessels very well. They have pulmonary hypertension. In utero, they get their oxygen by perfusing the placenta to get the oxygen from the mother's blood, also perfusing the placenta. So if they're born prematurely, say 1 or 2 kilos, their lungs are not developed. They shunt blood right to left through the patent foramen and patent ductus. They're hypoxic blue babies, and they don't do very well. They have a low APGAR score, and many of them die. If you give them low concentrations of nitric oxide inhalation, the blood vessels dilate, the shunting diminishes, and they survive and do very well. And that's really exciting for me to see that happen. And that all started back in the early 1990s. I was in Tunisia a few years back, and one of the Turkish neonatologists came up to me and said, "Thank you for discovering nitric oxide. It really is helping all my babies. However, it's very expensive." I said, "Nitric oxide's cheap. What's expensive is the machine, because you have to deliver just the right amount. If you don't deliver enough, it won't work. If you deliver too much, it can be toxic, because it gets oxidized to nitrogen dioxide, which is a toxic molecule." And I thought for a moment, and I said, "Why don't you give your babies some Viagra before you give them nitric oxide?" So now, many hospitals around the world give inhaled nitric oxide, and some are beginning to do it in combination with some Viagra to decrease the quantities of nitric oxide and the cost. But by understanding all of the biochemistry, it permits me to figure out how to treat various problems. And that's what I do. With my training, I combine clinical medicine with basic research. I just like to go back and forth. So there are a lot of other effects. Nitric oxide cycle at GFP. We know it regulates the differentiation of stem cells. It regulates platelet aggregation. Just does all kinds of things. Regulates gene expression, hormone secretion. Pretty remarkable stuff. 160,000 publications. I can't keep up with it anymore. I tried to, but it's impossible. I don't have that much research money. Thank you for your attention.
Let's put out a big hand for the top of the nitric oxide, Dr. Ferid Murad, our Nobel Prize laureate. And now, as the token of appreciation, we are going to really grateful for Doctor to come fly all the way from US to come here to give us the talk. Now, we are going to invite our founder of BEFIL to present this token of appreciation to Dr. Ferid Murad. Once again, to Dr. Ferid Murad, and thanks to our founder of BEFIL, Lee Xuesen. Okay, thank you very much. We're going to get going for the next program around. Another 10 seconds, yeah? I believe he has a lot of fans around the world. As being one of the Nobel Prize laureate, let's put a big warm welcome once again to our Dr. Ferid Murad. We feel grateful that in collaborations by our BE International with Dr. Ferid Murad for the formulations of the 360 Heart Health BEFIL of this product. And I'm sure everybody are here to get to know more about this awesome product with instead of L-arginine with the productions of NO. You can't resist the NO, no, but you have to get it. But before that, let me put to you, I'm sure a lot of people looking at it, I will just put it in more layman term for you to understand more about our BEFIL product. But before I bring him on the stage, let me tell you more about his background for our next speaker.